Hello again, everyone. So we are going to just do a sort of a brief rundown of what FAIR data are, what FAIR means, and we'll do sort of a run through of all the principles of FAIR, just a really brief overview. We'll give you some time to consider these principles kind of within specific context, and then we'll do a bit of a deeper dive into some of these principles and really sort of, I think, make more of the principles concrete through the different workflows that you can enable in your research. So this is just the primer, just something to kind of get our bearings on what FAIR means. So really, no matter what research you do, and that's really good to see all the different disciplines here today. And I'd like to welcome everyone again, and glad to see you. I think wherever you are in your career, and whatever discipline you're in, these principles are really applicable to you. The original FAIR standards are really easily found online. You can find those fairly easily. But the goal of this workshop is to really present these principles in a way that make the application in your current context accessible. So giving you a lot of examples. And also, you'll see if you look at the original FAIR principles that we're really simplifying the definitions as much as we can, just to make it, kind of confined within this 2-hour framework, and to make it as approachable as possible, no matter where you're coming from. So FAIR is an acronym for the principles indicating that data should be findable, accessible, interoperable, and reusable. Just first blush, these principles are obviously kind of highly interrelated, and there are multiple dependencies upon one another. For example, data really can't be reusable if it's first not findable. So, but I think the differences between some of these and how they're connected we'll sort of tease out here in a little bit. So the FAIR data principles really encourage researchers and other research stakeholders to ensure that data are findable just so that others can discover your data, accessible so that your data is available to others, interoperable so that your data can be integrated with other data or used by machines, and reusable so that your data can be used for new research, whether that's your new research or someone else's new research. So a little background on FAIR and where this all came from. They began as a workshop organized by a molecular biologist at the Lawrence Center in the Netherlands in 2014. And it's since been revised by different research communities. It's really become sort of this global best practice for data management. And the principles were drafted by researchers, funders, publishers, and university administrators who sought a way to improve the reuse of scientific data. And I think what's interesting about this is each representative in their original documentation really expressed their individual interests in FAIR data. Researchers just wanted better output to improve their scholarly impact. Funders wanted higher research integrity. Publishers wanted to improve the peer review process, more in-depth peer review of data. And university administrators wanted more publicly funded research and also greater impact and reputation. So they developed these principles with that in mind. And this sort of shared goal is really just more rigorous management and stewardship of digital data to the benefit of the entire academic community. The ethos of these principles is really that good data management and stewardship is not just a goal in and of itself, but rather a precondition supporting knowledge discovery and innovation. So that's really what FAIR principles are, what FAIR data is. And I just want to make sure that we talk about what fair data is not, and it does not necessarily mean that your data is open. It doesn't imply that you'll always share your data openly in a data repository. Open data is freely available and reused by anyone for any purpose. Fair data really provides a set of best practices for sharing data that respect the legal, the ethical, and contractual restrictions imposed by the nature of your research. So if your data contain sensitive information or subject to intellectual property rights, then you cannot share your data openly. I think fair data provides different approaches to making data fair, with or without these restrictions. So we'll discuss these principles more in detail later in the workshop. So this is all great, you know, that these folks have done this. It's kind of it's garnered global attention. There's a lot of interest, but why should we care? Why should we care about FAIR? And what's really in it for us? So I'm a proponent of being very nice and sharing data, but I'm not completely altruistic, you know. So I think we want to talk a little bit about first, you know, why we want to do this, not just because it's a standard or becoming more and more of part of you know, the requirements of our research data, but that it may actually have some benefit to you. So I'm going to start this off with another question for you. And I am going to talk about high impact research. We'll put this in the Google Doc as well. So this question is here. So what is high impact for you? What are the characteristics of high research impact in your field? So just enter your initials or just your response, initials and response, or just your response below in the Google Doc. And if you've just joined us, let me share that document one more time. Think about that. What is high impact research in your field? See, Heidi's already made an effort here. Easy to navigate, reproducible. What else? Everybody's still thinking. We'll give you a minute. What do you think? What are some of the things that you look at and think, this is really good research? Good. Anonymous Dragon is going for it. Thank you. Wide. I like that. Yeah, it has some sort of transferability to a wide circle of stakeholders. It's transparent. current technology practices, uses some standards. Great. These are great. Yeah. I mean, I was going to go with like the really low hanging fruit, which is it's just cited a lot. But everybody's been way more like thoughtful about it. That would have been my jump onto that one. I would have said citations, lots of citations. So if something's heavily cited. So yeah, I think, but these are really great examples. So thinking about that, and then thinking about how does FAIR make it more impactful? How does that elevate our research? And so I have a few examples here. So one of them is comes from this article. And it's really kind of a good explanation of how we can use data as a publication, as its own publication, as aside from our regular publications, our manuscripts. So really maximizing the output and the impact of a research project. So this is an article from Philosophical Transactions of the Royal Society. The article by Lombardi et al. Has on the left here, you see 1,758 downloads, very cool, but only 7 citations. But it was published in 2020, so that isn't surprising. If you look on the right, though, this is actually where a repository where they deposited their data, and where it's freely available online, and anyone can download it. You'll see that this screenshot shows that the data from their repository was already reused and cited 49 times. So the data actually has more citations than the paper itself. So looking at having very fair data allows us to increase our citation impact and make not only what people said about making it transparent, but I think there's even a sort of a connection just with how much more it can be cited and reused by other people who may not even need to cite the paper. So impact is one aspect of FAIR that can be very that one can leverage with FAIR data. This here is an example of reputation where data can go wrong. So this actually comes from Retraction Watch. This is a website that documents all of the different retractions that happen in scientific papers. This one, "We Are Living in Hell, Authors Retract Second Paper Due to Missing Raw Data." And sadly, this is one of many articles on that website about retractions from different journals. This is the International Journal of Cancer actually retracted these articles. The screenshot on the right is actually a study of retractions that looked at why papers are retracted and the reasons journals give. Their findings were not necessarily relevant to this, but I thought it was interesting that a lot of the reasons were that there's just incorrect data, there wasn't supporting data, it was inaccurate, it was misappropriated. So I think there's a real significant piece that's attached to our reputation as scholars, as researchers, for this transparency, that there's obviously the way that it can benefit us as a researcher, but if we're not sharing our data, if we're not making it if we're not making it open to others and transparent, then we may have issues down the line. And so that is one thing that I think helps to contextualize FAIR data and why we care about it, why one should want to at least explore aspects of the principles, maybe not adopt them all wholeheartedly, but at least apply some of them. So just a quick summary of this: they were developed by multiple research stakeholders to improve the research enterprise holistically. FAIR data are not always open data. FAIR principles emphasize sharing data with others, but these practices are beneficial to your scholarly impact and reputation. So we'll go on to kind of further defining FAIR data. This is really one of the first ones here, findable. One of the core pieces of this is to have a data set that should be assigned a globally unique, persistent, and resolvable identifier when deposited with the data repository. So when you deposit a data set with a repository, you provide some details about it. But the best part about it is that once it's in the repository, it will create this DOI. So this is actually an example of a researcher from engineering who deposited data and has now in the Texas data repository, our repository. And now that he's done that, the data set can be cited. So this is actually the citation it generates for you and includes a DOI, this persistent identifier. And we'll discuss these a little bit more in depth in all of these, but these are just some examples. So provide rich information about the data set, also known as metadata or data about your data. So this is a picture of the actual website, the repository where the data is located, and all of the information about the data in that repository. Data should be accessible. So this really is simple. That once you have that DOI, other people should be able to access it. So they should be able to at least know how to download it via a network URL whenever possible. And that's not to say that it absolutely has to be open and downloadable just like that on the web. But like I said earlier, access to your data set may need to be controlled, but this information should exist, and how it should be accessed should be available. So even if they can't just download it through that URL, how could they access it otherwise? And then we're looking at interoperable, means that data can be integrated with other data. So in order to make this possible, you need to ensure that you're using standard vocabularies that can be understood or at least referenced by others. So really ensuring that all the variables and the language that you're using with your data set are appropriate. And biomedical sciences actually is really great at vocabularies and ontologies. They really are, they love an ontology. And so they're excellent at documenting them too. So this is on the right, an example of a database of ontologies. So that's right, it's a database of databases of different ways we name and group biomedical language. So, so there's always a way to find out if you are naming your variables correctly. Interoperable also means that the format in which you encapsulate your data can be opened and transferred without significant labor. So using open formats such as text files or CSV is preferred over proprietary information such as Word and Excel. So in some cases, you'll be working with proprietary or instrument readings that export in very specific formats, but there's often a way to transform that into an open format that others could use. Interoperable also means that that our data are and metadata are human and machine readable. So machine readable is really data is in a format that can be easily processed by a computer without human intervention, while ensuring no semantic meaning or loss, so no data loss. And so really, I think kind of another way to define this or describe this is that the machine readable test, if you will, is any structured data or data that you can copy and paste and can be accessed using a URL tends to be machine readable. Right, we'll move on to reusable. So reusable, again, depends on all of the other applications of FAIR. But information about the collection or the generation, this all document, data encoder accompanied by documentation that describes context about your data materials. And your documentation, we saw a lot of people here are using README files. That is absolutely something that makes your data FAIR. That documentation just is a short list that we'll dive into a little bit more, but title, author, date of collection, geographic location, methodological information. And your documentation can take the form of just really well-described file names, that README text, code books, or if you're using any code, using some markdown to describe your coding process and your methods. And really, this documentation is just the who, what, when, where, and why of the data. It can be described in many different ways. So for this section, we just thinking about these principles, they really can be integrated throughout the research lifecycle, and they're really highly interconnected. So it's not like you're just you're working on your data, you're working in your lab, and then at the end you make your data FAIR. A lot of this documentation, the interoperability, really happens at the beginning. And it's really helpful to consider your goals for sharing the data as soon as you begin collecting it. So thinking about where you'd want to put it so that it can have a DOI associated with it would be really helpful. So this leaves us to our big breakout session, our first breakout session. So we're going to break you up into groups of 3 or 4, depending, and we will have we'll actually have each group, this will be on our Google Doc as well. Let me just bring that over really quickly. What we'd like you to do is, as a group, choose one of the data sets. So these are all data sets that are available in a repository of some type. So you can actually see what it looks like. Put that in there. And this is, so as step 1, as a group, you'll choose one data set from the list below. So you have a choice of any one of these 4. Just choose one person to take notes as you go. I'm going to copy that in too. I have some prompts for that. And then just answer the one question that we have below your group. So your breakout room number will correspond to your group number. So you'll see which breakout room you're in, and then you will just answer this one question below, which is, knowing what you know so far about FAIR data, is it fair in this data set that we've given you? Why and why not? And there's really no binary here. It can be a little bit of both. But I also, just because, we're not totally mean, giving you this little primer and then making you do it yourself, but we do have a link to the FAIR checklist, and that is right here. So this is just a little bit of the bullet points of what we've already discussed. So go through this checklist and decide as a group what you think, and make sure that you're taking notes. This is kind of how we'll know that the groups are ready. So I think unless there are any questions, and if you haven't, maybe if you have the Google Doc up, you may need to refresh it. If everybody is having any issues seeing the Google Doc, but I know that sometimes today I've had some issues with it not being able to refresh. So if there aren't any questions, we'll go ahead and launch the breakout rooms. And we'll give you about 20 minutes for this exercise. Okay. Carly and Diego, are you able to join your rooms?
I was able to join. I'm just monitoring. I don't know if I could really add much to the conversation.
Oh, you're fine. I wasn't sure. Okay. Is it okay if we pause the recording while we're in the breakouts?
Yes.
Okay, I'll do it. I could do that. Thank you. I need a reminder sometimes. Okay, I think we're all back. Welcome back. All right. So I saw lots of activity happening in there, and your notes are really thorough here. And I know some of you are still finishing up, and that's more than fine. It looks like both groups did the same data set, fun, and it was a good one to do. And I mean, your thoughts are in here, and I couldn't agree with you more about the oddness of the link for the data is included as part of the results in the paper, but the raw data is the data set and but it's really kind of a bizarre web page. The data sets aren't labeled clearly, and the abbreviations to understand the labels are at the end of the paper and not included with the data set in a README file, which is also, yeah, it's, I think, a really good insight, very specific, and absolutely something that, you know, I think a lot of people will interconnect the paper and the data and kind of rely on the paper to explain a lot of the data, but it's much more useful and fair if it's connected within, or actually embedded in the data set in a README file. And we had group one saying no direct website linked to the data set from the page of the journal. It's sort of buried. I agree with that. And that the accessibility piece doesn't necessarily fit the principles of FAIR. So I think that's great. So just and sometimes it can be you know, something, and I think this is the case. I think if you think about fairness as a spectrum, sometimes we're not going to be able to implement all of our FAIR principles into our data, but we can include some. So, you know, something is better than nothing. That's especially the rule of thumb when it comes to documentation. So we'll move on. Thank you for taking a minute to just look that over, get some practice with that, kind of with a fresh data set, a fresh pair of eyes. And we'll kind of take this time now to dive into workflows that enable FAIR. And we'll start with planning and documentation. So this is actually things that you can implement in your research workflow to enable FAIR data at the end of your research life cycle. So Daniel, take it away.
For sure. Do you want to share the slide deck? Or am I missing it somewhere?
Can you see it? Can everybody see this?
No, I see a very nice image of your face, but not the slide.
Oh, interesting.
Not the slide. That's okay.
I wonder what happened. Let me see. Oh, I know what happened. Okay, we're here.
Awesome, perfect. Thanks, John. Yeah, so as John was saying, we're kind of scoping out a little bit now. Basically, the activity that we just did was you were looking at the data sort of associated with single papers or, you know, single events, single DOIs. And so pulling back into workflows and planning, this is more thinking about universal principles to apply to all of the kinds of projects that you work on. So basically, if you or a PI that you're working with or someone like that is submitting a grant to a federal agency these days, essentially all of them are going to require a data management plan associated with the data that you're going to generate. through the course of the proposed research project. And well, just as a junior faculty member, my big piece of advice here is don't see this as like busy work, as something that you have to check off as a box, but instead use this as an opportunity to really think critically about planning your research, maybe from a slightly different direction than the pure research description is. So we'll talk a little bit on subsequent slides about how one breaks this down. But one thing that I would definitely recommend is looking at the links for the advice that Texas A&M has on putting together these documents. And I'll go ahead and just paste those links in the chat right now. So the first one is a general guide on data management plans that the university puts out. And most universities' vice presidents for research offices will also have similar guidelines. And so I think ours is pretty good, but you can always Google, data management plan tips and get some pretty good resources on that. I also recommend that you talk with your colleagues on sort of seeing how they frame their data management plans. That was what I did when I was first starting out writing these. But always use that as a starting point and think critically about the kinds of data that you'll be generating and sort of to what extent you can share it. We all wish that we were like the astronomers, where all of our data could be as open as telescope data is all the time. But I, as a material scientist, or you as a biomedical oriented person, have different interests than just purely sharing deep field universe data. And one particular consideration that you should always consider is how this is going to fit in with the sort of IRB protocols that are present with the kind of data that you're sharing. So I think that we're actually ready for the next slide. So one way to think about writing a document or coming up with a data management plan is to break things down in this way. I think that this is a pretty effective one because I think it naturally flows out of how one designs a research project. So you can think about the kinds of methods that you'll be using for your project. And I'm a computational chemist, so I don't have to think so much about instruments, but many of my experimental colleagues do. And it's at the instrument level that you start thinking about proprietary data formats. What are your protocols going to be for converting those data formats? Does it just produce terabytes and terabytes of data that you have to think about aggregating or filtering out? And to what extent, do your facilities and equipment document fit in with actually being able to handle the data that you're generating? Instruments and software are somewhat interrelated in that way. Most instruments are operated typically by proprietary software these days. And then finally, the one way to think about this is you could either divide this document into headers associated with these ideas, or you could think about having a dedicated section for how you're going to disseminate the data. And when you think about how you're going to disseminate the data, it really depends on the various stakeholders that are present in the project. And you yourself are a stakeholder, but you should also think about the interests of any collaborators or co-PIs or students that are associated with the project, any private agencies that are associated with it, and then of course, funding agencies and their standards for funding it. This could become particularly important whenever you have NIH funded research, because there are very specific guidelines on making both your data and your publications available. And then finally, you could consider for really big projects, developing repositories or hooking up with existing state-of-the-art. And I should point out that I've been on NSF panels to date, And people really do read the data management plan. And there's in fact a dedicated section to commenting on the appropriateness and quality of the data management plan. So it really can be a big benefit to how creating a cohesive narrative around what you're doing, or it can be a big detriment to going on what you're doing. So now I think Laura is going to talk about metadata. Oh, Laura, you're muted. There we go.
I am muted. Next slide, please. So let's talk about metadata. This is just data about your data so that it can be understood by others. It includes basic information such as the title of your dataset file, the meaning of variables or abbreviations, and how and when the data was collected, analyzed, or anything else of use. Next slide, please. So you are already creating metadata in your everyday life. Say you take a selfie in front of Evan's library. Your phone automatically creates metadata such as the GPS location, the date, time, file size, file format, and information about the camera. Then if you post this on your social media account, you may even add more metadata like a Twitter hashtag. Next slide, please. This slide shows how metadata can be helpful to others. If we look on the can on the left, it has no label. We assume that there's something in the can, but we don't know if it's food or paint or glue. This is the equivalent of just handing somebody your data spreadsheet with no background information or metadata. In the middle photo, we can see the can's label has metadata that shows this is tomato soup. So we now know it's an edible product. The label also includes metadata that shows the brand, the food type, the food flavor, and the size. But when you provide rich metadata, such as the label on the can on the right, there is information on the contents of the soup so that now I can decide if I want to eat this can of soup, or if I look closely at the amount of sodium, opt for the healthy version with low sodium. So by providing detailed metadata, you help others decide if they want to use the data you shared. And in the future, you don't have to try to remember how your data was collected and organized if you have good metadata documentation. Next slide. There are specific metadata standards for biomed. The MESH vocabulary system is the thesaurus used by the National Library of Medicine to index articles for PubMed. By using a common vocabulary, we meet the I in FAIR to be interoperable. Below are a couple of links that are examples of NIH programs that have their own metadata standards or best practices for their specific discipline. One example is the DMID, Division of Microbiology and Infectious Diseases, that calls for details about how the preparation of DNA samples for sequencing was used and any other protocols done prior to sequencing. So check out your specific discipline and see if they have metadata standards and best practices. Next slide, please. So after looking at these metadata standards, what kind of information would you need to reuse the data that you reviewed in our earlier exercise? You can just go ahead and put these in the chat. I think I see some people typing. So was there any metadata that you thought should have been included in that data set that you reviewed that would have helped you reuse it? Because I know one group said that there was lacking metadata describing the data. So is there any specific information that you wished was in there? Okay, well, I guess I'll go ahead and move on. Next slide, please. Maybe people are a little tired after lunch. So now we'll look at data workflows that enable FAIR formats. Next slide. Oh, thank you, Daniel. Daniel is pointing out that metadata is not as simple for some fields as it is for photos, and that's true. So let's look at data formats. Use data formats that can be accessed easily by anyone without contacting you for help. On the right is an image of a PDF that shows data. It is a good visualization, but it is not helpful for someone wanting to reuse the data. So to be fair, it would be better shared in a spreadsheet form as well. Also, as John mentioned earlier, use open software rather than proprietary software so that other users don't have to purchase high-priced specialty software to read your data. Next slide. Here we have a list of proprietary formats and their open equivalencies. Notice again that Microsoft Office is proprietary. Also with open formats, you don't have to upgrade files. Like when I had to upgrade all of my XLS extensions to the XLSX extension in order to ensure that they function properly. Next slide, please. And I'm sorry. Next slide, please. Finally, to make your data meet the A in FAIR, you can dictate how your data can be accessed and used. When you create your metadata, you can add this information and use licensing, such as the Creative Commons licensing image you see in the corner. The image explains to people that they can use your data if they cite you, but they cannot use it for commercial purposes. So now I will turn it over to John.
Right. So we'll do a little bit more about persistent IDs, and I will say the FAIR principles are really heavy on persistent IDs, as I think most of the research landscape is as well. So I think the persistent ID or a DOI that we see in our articles has become the gold standard for for having a permanent place, a permanent home for any digital object that we place online. So the persistent ID is just a long-lasting reference to a digital resource. And we have our DOI, digital object identifier, such as journal articles, books, and data sets, but you can have many, many more different types of PIDs or persistent identifiers that will provide that ongoing and sort of curative preservation of your online content. So we'll go back to our example here. This is, again, a picture of the data repository from this researcher here at Texas A&M University. And we already showed that we deposited the data in the repository, received a DOI for that data here. So we have that DOI, but when we think about our sort of scholarly ecosystem and how our work can and should be interconnected. And if we are thinking about data as another citable, publishable product, then you have your DOI for your data, but then you're able then with that DOI to track your metrics and citations for the data. And you're also, as some people noted in the data set, that you're able to link the data from your publications and then also link your publication from your data set. So you have that connectivity between your products, making your, well, it's very satisfying as like a neat ecosystem for your data, but it also allows your stakeholders, as Daniel was mentioning, to find all of the results, the products, the impact of whatever they funded, your university for promotion. So these persistent IDs are really key to that findability, the F in FAIR. And I think the easiest way to approach this persistent ID is making sure that data is available in a repository. A lot of people obviously put data in GitHub. GitHub has the ability to mint a DOI for your data. However, you must actually transfer the data to a repository in order to receive that DOI. So it links it to a Zenodo repository. So people share often in GitHub, but GitHub does not give you that snapshot of the data as it was when you reach the results for a paper that you've written. That's how did you answer your research question, and how did these data support your answer? So that conclusion is supported by data, and that data is connected then to that paper. I think persistent IDs are also very important so that we don't get that lovely 404 that we often get when we are clicking through links from really old papers. If you've ever gone through a research paper from the 90s, early 2000s, and tried to find another resource that's linked within, the 404, we call that link rot, and it's definitely very frustrating for anyone who would like to connect that paper with the actual data. When you have a repository with a persistent ID, you have what's known as a tombstone page. So that persistent ID will live on forever, even if the data is deaccessioned. So if someone takes the data away, in this example on the right here, this public use microdata sample, the data was removed, but the actual page remains. So, and they would be able to contact whoever was the author of that paper and that data, and it often gives information about the data set to say, yes, this did at one time exist, and maybe it was moved, maybe it's in a different repository. So that's the other, I think, the pro of keeping your data in a repository with a persistent ID. I do have an example here of an article that was written in 2009. I'm actually just going to link out to this really quickly. So this was actually an article about public sharing of research data sets and looking at all of the sharing practices of other researchers. If you look in their appendix, they have supplementary data. And you can actually download the data as a text file from the paper, but there's no persistent ID. And as a matter of fact, this is a fairly new, they revised this. I guess maybe the journal allowed them to, but for a while it was a link that ended in a 404. But now they've embedded the text, but there's still no persistent ID. And I think what happened was this was written in 2009, maybe before they had access to a repository, or this became a standard practice for these authors. So they did deposit the data in a data repository later in 2011, but the two aren't interconnected. So that's, I think, one of the issues when we're not planning ahead, like Daniel mentioned, to really make sure we know what our end goal is for the data. If you're not ready to share that persistent ID from the repository in your paper at the time of review, it may not make it in there. So others won't be able to find it as easily as they would if it was embedded in that paper, or at least somewhere on the website. So, which was also an issue that our groups 1 and 3 found with our example data set. So, so you'll see this was 2011. So I think they thought, oops, we should probably put this in a repository at that point, but they did miss out on that persistent ID in the journal. So a summary of this idea of kind of everything we've gone through in terms of our workflow is really make sure that you're doing some work at the very beginning of your research life cycle, really thinking through the FAIR principles, and in order to make your life easier at that publishing and sharing results stage of research. And that will keep you, I think, from some of these data danger zones that we find with FAIR data. So I know we're working with some folks in biomed. Some people may be working with sensitive data. So we wanted to make sure that we explained sort of what FAIR means in the context of sensitive data, and making sure that if you are working with subjects, human subjects, that there are ways to make data fair, but there are also many obligations on researchers when attempting to do so. So just to go through some really briefly, just some definitions here, we're talking about PII, personal identifiable information, any representation of information that permits the identity of an individual to whom the information applies to be reasonably inferred by either direct or indirect means. So we'll talk a little bit more about what direct and indirect mean in a minute. So that's my personal information. And then we have protected health information, PHI, and that is the HIPAA privacy rule, federal protections for personal health, making sure that our privacy is enabled whenever anyone in healthcare is working with our health information. And it does sort of have some flexibility with it, but there are a lot of stipulations that come with this federal regulation. So there are a lot of demands on us, but what a direct measure or a direct identifier is, can be any one of these here: the internet protocol address, names, social security numbers, your medical record numbers, phone number, your address, certificate number, or license number. So really any unique identifying number or code that's associated with a person. That would be a direct measure. I couldn't make this as visual. It was too detailed, but indirect would be not necessarily this code or a set of ID numbers, but detailed geographic information. So if I have a little bit of information about someone and I know where they are in a sort of a state, a country, then I miss, or especially a county, a city, I can make some connections between their ID and this location. If there's any specific organizations that they belong to, those can be traced back to an individual. Detailed occupational titles can often narrow down a pool of people, make them identifiable. Exact dates of events. So if you're giving birth dates is a big one, death, marriage, divorce, and any detailed income information can kind of connect indirectly to an individual and their personal information. So there's some ways to remove data and protect some of this these direct and indirect identifiers. I have some protection terms here, so you'll find that these terms have some overlap, and in different contexts they may be used interchangeably, but we'll just use these definitions for today's purpose. So looking at, there's the option of anonymization, so that's all linkages to an individual are eliminated or manipulated to the point that it's that it is not at all re-identifiable. Let's say that you need to retain individually in your data, you'll need to apply some technology to data points to irreversibly prevent identification. No one, even the original study organizers, can re-identify the participants under any condition. It's truly anonymous. De-identification, all of the direct and indirect identifiers are removed, but it's possible, perhaps, using some sort of key or link back if needed to identify those participants. And then pseudonymization can either be like replacing names with John Doe, or it can be like using a student ID instead of a social security number. In the John Doe instance, you can sort of figure out who it was originally, but the student ID is part of a key that can relink to the student. So there still exists in pseudonymization some linkage back to that individual. So I think you can definitely see that there's overlap between all of these, and there's some interchangeability, and they are somewhat coextensive, but these are different methods one could take to protect individuals if you are using human subjects and if you wanted to share your data openly with others. So, and there are several methods of protection that you can engage in. In some cases, the ability to re-identify is desired by the identifying body. So the researcher may want to go back. Another layer of risk sort of lies within the potential of a malicious party to access the data linking directly to that. So in anonymizing your data, you need to kind of completely remove all direct and indirect identifiers, remove those links to external data. De-identification, recode those geographic areas, resort and renumber cases from original. And then if you have a number of variables that are very specific about the individual, create ranges for those variables. For pseudonymization, create pseudonyms using a key, hash, or code, and linked to a database with the original data retained securely. So you can share the data once it's gone through any of these protection methods, potentially, depending on the legal ramifications of any kind of contract that you have for the data. There are some examples of shared de-identificated data. So this one is from the Veterans Affairs Precision Oncology Data Repository. So this was a repository of veteran clinical genomic and imaging research that was actually all available in our repository, but completely de-identified from any of the subjects. So that's a full repository of data that anyone can use without linking back to the individuals that they were studying. So it is done. It is kind of difficult to find data that is sensitive in this way, open and somewhat fair. Well, what if you can't? So what if it's just too much to handle, and you say, okay, we're not sharing the data openly, but what could we do instead? And to make this maybe even not the data fair, but elements of the project fair. So creating more of an open science pipeline, you could only share metadata about the data. So, and some contact information for acquiring the data set, given that they use some sort of data agreement. These other researchers, you can still share instruments and surveys in different repositories. Share your protocols. I have an example of that. Share your software code in a repository. And of course, any of our aggregated data and methods for aggregation could be shared. So not the actual data set, but all of the connected sort of methods and work around that materials. So one example of this is this shared protocol. This is in protocols.io. It actually is an open source product that comes out of Berkeley. And just click on this really quickly. This is a protocol that these researchers have shared. I also really like that. I think it's a panda. No, it's a, what is that? Is that a raccoon? This is their icon. And they have, they've shared this in association with their paper. On this is essentially just a repository for protocols. So they have all of the authors here, and they've gone through and actually written out all of the steps for this protocol on illumination power and illumination stability. So they've detailed this to great length and shared this openly. This is their entire protocol here. What's really nice about a platform like this is you're also given a DOI, that's right here. And so someone can cite this. This also has some sort of social media aspects to it, so people can comment. Someone said, great work over here. And then it also, people will test it and say whether or not it worked for them as well. So you have a peer review of this. And so they have 23 people voting that. So also another great way to look at your metrics from your research products. So this is just one example of other ways to make your research fair, even if it doesn't include any sensitive data that you absolutely can't make available. Okay, so we're going to go into a breakout. Again, this one, and we're just going to go into our same groups, and we'll go again, choosing one person to take notes as a group. For the group, as a group, we'll review the following resources, or the resource for identifying. So these are actually, I put in two just in case. So there's one that's kind of a basic steps for de-identifying data. And then there's one that's like a little bit more advanced. So if you're really feeling like you wanna take a risk, go for it. But we'll have a couple of questions in the Google Doc for you to answer as a group. And we'll copy those in as well. So for this one, I think we have about 15 minutes. Let me grab those. And I will paste it into our document. Okay. So again, we'll set you free into the groups, and we'll have our notes for the three, and we'll have about 15 minutes to discuss these. So just pick whatever one you'd like, and then we'll take and try to find someone to take notes for that group, and we will meet back in about 15 minutes. Any questions about this breakout? Now I'm missing my two other groups here. Any questions? No? Okay. All right. See you in there. Oops. Okay. I think everyone's back. Welcome back. Everyone was so busy typing. It's good to see. Yeah, I thought everything that everyone said looks really great. It looks like we had group one looking at masking, some randomization, and then talking a little bit about how indirect variables linked to external data sets are challenging both to locate and remove the risk, which is really tricky if you're using a lot of, I mean, our information is linked everywhere. So it's really hard to disconnect those links. So that's really true. And both groups talked about consulting a professional. I think group three also talked about masking, randomization, removing, replacing. It's definitely not as straightforward, and definitely I think it's good to consult with someone who has that expertise. There are a lot of tools in those resources for larger data sets if you're working with big data. For how to do that, I know it can be really difficult to just sort of randomize something, and you can do it pretty easily in a spreadsheet, but what if you're working with terabytes? So there's definitely software out there that can help you with that. And I think both people talked about, if we can't share the data set, sharing the metadata or information about it and contact information makes that data fair. So that leaves us finally to we're closing. Oops. Hang on. What happened? There we go. Nope. Our reflection. Daniel, take it away.
Yeah, so just to wrap up today, we always like to close these kinds of workshops with reflecting on some of the things that you've now gained, because we sort of had our pre-reflection where you talked about what principles you were already using or learning about. And so instead of sending you right back into breakout rooms as a group to discuss this reflection, instead what we'll do is we'll have you type on the Google Doc an answer to one or two of the following questions. And so, for instance, what could happen that would enable you to feel fully engaged and energized about fair data? There's a lot of discussions still in this community about proper incentives for fair data. And often people from one field, their incentives will be different than people from other fields. The second pair of questions is sort of what's emerging here from you, like what are the things that are really sticking out, and what new connections are you making between sort of the fields of research or biomedical data that you're interested in, and these principles. And then finally is the most pragmatic of all of them is what would it take for you to make your data fair? And perhaps that could also involve how much effort would it require? And that might help you think about when you're starting your next project, front loading some of that effort, like John talked about earlier. So we'll just give you a few minutes to sort of pick one or two of these questions, but feel free to copy them over and think about them long-term as well.
I'll also say, if you can't answer all of them, that's fine. One or two is probably good enough. Ooh, more training on metadata standards. I'm a fan.
It's definitely something for PIs and co-PIs to think about when they bring in new graduate students or researchers in general into their groups. Having sort of a standard operating procedure for these kinds of things.
Mm-hmm. I know that we're at time. I know if Christy, did you want to say anything?
I just had some quick closing remarks whenever you all are finished.
If you want me to stop sharing my screen,
If you'd like.
great. I can stop.
Okay. So I just want to thank you all for joining us today, as well as our presenters. Just as a reminder, our next session is going to be Wednesday, April 6th, same time, 1 to 3 p.m. And this same group, Drs. Watts, Sare, and Tabor will be our presenters again, and they will be discussing FAIR in the real world. Thank you again.
All right. Thanks, everyone. Appreciate your time, and we'll hopefully see you in April.
Yeah, those were some great thoughts and answers. And I know it was a lot of information, but it'll be recorded, so you can always go back and think more about it in the future.